This is the final seminar in our spring seminar series for the Pacific Southwest Center of Excellence in Vector-Borne Diseases. So thank you all for joining. And I know many of you are starting to get busy with at least mosquito season. And summer is here academically as well. We're in finals week here at UC Davis, and I presume that's true at Irvine as well. So first up, we have the pleasure of having Gaëlle Sehi, who's a fourth-year PhD student in public health at UC Irvine. Her work is focusing on spatial epidemiology and ecological modeling. Her current research uses environmental data and machine learning to predict the abundance of Aedes aegypti mosquitoes in Southern California. She's collaborating with local mosquito control in the area. And her work is aiming to inform vector surveillance and control strategies. Broadly, she's interested in access to healthcare, particularly in West Africa, and how spatial data and predictive tools can be used to address disparities in health systems, disease prevention, and resource allocation. So with that, I'll turn it over to you, Gaëlle.
Thank you so much for the introduction, Chris. Good morning or good afternoon, everyone. It's really good to be here and be able to present my work. Okay. Well, so today I will be talking about ecological model and how we use this ecological model to predict Aedes aegypti abundance in San Bernardino County. So this is, okay. So this is a summary slide of the work that I'm doing. For the past 8 years or so, the number of mosquito has been increasing in San Bernardino, and that led to us wondering why. And we hypothesized that environmental factors like temperature, urbanization, and water, so precipitation and irrigation, so water retention, are factors associated with the abundance of mosquito. And to test this hypothesis, we downloaded satellite images from using Google Earth Engine, and then overlaid the count of mosquito, and then implemented all of those variables into ecological models to better understand the increasing number of mosquito in the study area. So the mosquito that I'm studying is Aedes aegypti. The mosquito is common in tropical and subtropical area. The mosquito is known to be a daytime biter, an urban dwelling mosquito, and a vector for diseases like yellow fever, Zika, and dengue. So when we talk about dengue, a quick example of the global burden of the distribution of dengue. When we think of Aedes-borne diseases in the world, they're still underdiagnosed, and surveillance is often lacking in different parts of the world. This map is the prevalence rate dengue cases per 100,000 population from 2024 to 2025. I obtained this map from the CDC Europe, and showed where the cases were detected in the past 12 months. And the prevalence rate were higher in tropical countries like in Asia, in Africa, and South America, Central America. And the prevalence rate are not high in the United States, but the United States are at risk for higher or increasing number of incidents due to various reasons like climate change, so increasing temperature, increasing rainfall, so more appropriate climate and environment for the mosquito that is invasive in the United States to expand. So as a public health expert, I guess it's also good for me to talk about dengue. And dengue is a vector, well, a viral infection. And most people who do get dengue do not have any symptoms. But when people, the most common symptoms are high fever, headache, body aches, nausea, and rashes. And usually people get better after a couple of days, but in rare instances, people will develop severe dengue, and that's when they will require hospital care, or in those instances, the disease can also be fatal. As of now, there are no cure or treatment well, they're not cure for treatment. So dengue is usually treated with pain medicine and a lot of rest. And um, and yeah, and all of those information and background led to the work that I'm now doing here in California, which started with the creation of an ecological model to better understand the expansion of the invasive mosquito here in California. So the study area is in Southern California. It's in the dashed area right here near Los Angeles, Orange Riverside. And and well, San Diego is not near, but not far from San Diego. And the study area is the West Valley region, San Bernardino County. This region has experienced increasing number of housing and housing expansion and construction due to the high cost of living in Los Angeles. And we also hypothesize that those human movements and creation of housing could be factors also associated with the increasing number of mosquito. So all of the traps are or will be, all of the traps were installed in the study area that's right here. And how is the data collected? The data is collected by the West Valley Vector Control District. They have an amazing team there where they install BG Sentinel traps by residential area, parks, and with the approval of the homeowner, they leave those traps overnight and then collect them in the morning. And here there is a quick shot of mosquito traps, very active in the morning, still looking for a blood meal. And they bring those traps and mosquito back to their offices, and then they count and identify the mosquito that they have. So, and that's where I kind of that's where I play a role, and I map and I use this data to better understand the disease ecology of mosquito and population in this study area. So this map shows the distribution of mosquito caught from 2017 to 2023. The dots are the counts per trap per night of mosquito for each year, and mapping the counts per trap per night allow us to observe the trend in space and time. That's just a visual representation of the mosquito caught from 2017 to 2023. And what we observe is that the abundance of Aedes aegypti has increased in the study area and over time. And even when we account for the trapping and surveillance effort, there is still an increasing number of mosquito. So in 2017, it's the very first map right here, 358 mosquito were caught compared to 2023, where we notice more red dots, so increasing number of mosquito. And in total, 30,147 mosquito were caught during that year. So just visually, we observe that the number of mosquito caught over time is increasing. And with that, led to the very first research question, which was to understand any spatial patterns of mosquito in space and over time. And that's where we use our spatial analysis, spatial statistic. We employ a Geddes Ord statistic to identify significant local cluster of high, so the hotspots in red, and low cold spots in blue of Aedes aegypti abundance. We applied a fixed distance spatial weights matrix with a 500-meter threshold, and that was informed from a correlagram analysis that we previously employed to determine the local clusters of mosquito. And what we observe with the spatial analysis is that in the earlier years, hotspots were concentrated in the south. So in 2017, we observed hotspots right here near Chino Hills Park. And then the hotspots persisted in 2018, but the other hotspots then appeared in the west part of the study area and then expanded. But the same hotspots near Chino Hills, these have now become a cold spot. So that could also be the reflection of the control and surveillance techniques used by the vector control district over at the West Valley. But those hotspots also move in the later years. They're either disappeared or moved now towards the north of the study region. And now we're just looking at the spatial patterns of the mosquito. We haven't even taken into consideration the environmental or meteorological factors that we hypothesize could be associated with the increasing number of mosquito. So the research question was, what factors are driving these patterns? And I will jump straight into the model that we use. Before even using a model, we kind of use a framework, an ecological framework. It's a visualization to pretty much show the association between each covariate, but also how each covariate could be impacting the outcome. Here will be the mosquito abundance. And we took into consideration all of those environmental factors impact the biology and cycles of the mosquito in different ways. So we hypothesized that the spatial and temporal distribution of mosquito was positively associated with precipitation, surface water, and built environments. We also hypothesized that higher temperature will be associated with higher mosquito abundance. And also have location here because when we think of this ecology or even any organism, it's good to keep in mind that the geographic area could be specific to that environment. So in a tropical environment, would have included humidity and vegetation. But because the climate here in Los Angeles is, in Southern California is arid, we did not find that it will make sense to include those variables. So with those environmental factors, we extracted precipitation, surface water, and average temperature with a 14 and 28-day lags from the mosquito collection dates to account for the time needed for these environmental factors to influence mosquito development and population dynamic. And then implemented all of those covariates and outcomes into a general additive model. I will zoom in on the very first two graphs right here, but a quick overview is that we use a negative binomial generalized additive model, and the GAM allow us to account for the complex and potential nonlinear association between the environmental variables and the outcome here, which is the count of mosquito. And we found that surface water, which is B, has a positive association. Surface water has a positive association with the counts of mosquito, as well as average temperature. We also looked at the interaction between average temperature and surface water onto the effect on abundance, and noticed that those two variables, when interacting, also have a positive association onto the abundance of mosquito. When we looked at elevation, urban area, precipitation, we noticed that those variables did have minimal effects into the abundance of mosquito. And same as with precipitation, has a negative association. And then taking a closer look at the first two graphs, is that the very first one on the left is a spline interaction term for latitude and longitude, and is a representation of the association between location and abundance of mosquito after accounting for the other variable in the model. And this map demonstrates a clustering of mosquito abundance in the south and in the west of the study area. So the colors here are inverse. So wherever there is a bright yellow, that's where we notice clusters of mosquito. And then taking a closer look at the surface water, I think that one was one of the most interesting graph that I had and trying to understand what was happening. But in overall, there is a positive association with abundance of mosquito, as we notice right here. But then once the surface level becomes high, we then notice a negative association, and that could be the reflection of water flushing the breeding sites away. And so with this model, we were able to, so using this ecological model, we were able to determine that factors like seasonal temperature, human activities, and surface water were factors impacting the increasing number of mosquito. And using that same model, we were then able to further think of prediction of the mosquito in 2023. So prediction or forecasting is now used oftentimes in disease ecology, and forecasting is using past patterns and current environmental data to estimate future mosquito abundance, where we can predict short terms, which will be week in advance, or we can also predict long terms with more like the seasonal trends and year. So previous literature have used prediction in different states in the United States. So warmer states like Texas, Arizona, Los Angeles, and in Miami, but also in Europe, where the mosquitoes are now becoming, invasive species. And mosquito controls operation represent the main means to prevent and control local outbreaks. However, the majority of the vector controls in the United States are in need of improvement. So it becomes important to develop an analytical tool that provides real-time forecasts for mosquito relative abundance. And that's where our work comes into play. And knowing all of those information, we wonder if we could accurately predict the abundance of mosquito in 2023. So our prediction work is divided into three parts, where the same data that we use for the construction of the ecological model, we then divided into training and testing. So training is 2017 to 2022 that we will feed into the models, and then use the testing data 2023 to observe how the prediction are performing compared to actual data. So once we feed this training data into the models, we use the exact same generative model that we created in the very first part, and then use random forest machine learning that is widely used for disease ecology. But one downfall or limitation of random forest, although very good, the limitation is that the spatial component is still I wouldn't say lacking, but there is still room for improvement. So what we did right here is that we created a third model still using random forest, but now taking into consideration spatial autocorrelation. So the hot spots and cold spots that we observed earlier are not so much taken into consideration in the random forest. And with those models implementing now the data, we obtained two different outcomes, so a spatial and temporal prediction of the mosquito in 2023, and then use the actual data as a counterfact to observe the performance of those three models. And moving on into the temporal prediction, so the blue line is the observed data, the 2023. The green one is a generative model, and the two others, so the orange is a spatial random forest, and the red one is a simple random forest. We noticed that all three models pick up on the seasonal patterns of the mosquito, so they all long-term predict the seasonal patterns of the mosquito. However, the generative model over predicts the abundance of mosquito, whereas the two random forests although very close to the observed data, underpredicts the abundance of mosquito. And then moving on into the spatial prediction, the general additive model is predicting the abundance of mosquito well, where we notice a higher number in the south and west region of the study area. And like I mentioned earlier, random forest still has limitation when it comes to spatial prediction. So in the middle, not much was being observed. And then when we add the spatial autocorrelation components into the model, we then pick up on finer details or finer scales of that prediction. And in summary, we were able to build an ecological model using environmental variables to predict the abundance of mosquito. And we found that high mosquito abundance were associated with warm temperatures, so in the summer, and moderate surface water and areas with higher population density were also associated with high mosquito abundance. We also were able to identify the strong seasonal patterns with peak in the late summer and then hotspots in densely populated area. This matters because the model supports targeted vector control in time and space. It helps anticipate future outbreaks and optimize intervention, and hopefully it can contribute to data-driven mosquito surveillance strategy. This work will not be possible without the help and the funding of PacVec, and will totally not be possible without the support and help and data from the West Valley Mosquito and Vector Control District, and the support of my advisor and people here at UCI. So thank you so much for your help and support. And I will take, I'm not sure if we're doing questions at the end, but I will take any questions if you have any. Thank you.
Thanks, Gaëlle. Really great talk. And I think what we should do is take a few questions now, if there are any, just before people forget, and then we'll move on to Dr. Attardo a bit later. Does anyone have questions or comments for Gaëlle? So I'll start with one. I was curious about, I'm accustomed to models generally under predicting, because usually I think you're modeling on a log scale, so to speak, right?
Yes.
And I'm kind of surprised sometimes they over predict, but also they seem not to be capturing the exact shape of the observed data. The curve you showed had the spike in the blue line for the observed, but it seems like both of the random forest models, sort of reach a peak and then slowly decline, or I'm not sure what's happening with the spatial one, but do you know why that is happening?
I think with the random forest, I also noticed that there is still, there is this weird shape at the end where, that's a really good question, and I wouldn't know how to answer it.
Seems surprising, especially in November. I think almost all parts of our state are dropping pretty dramatically for Egypti.
Exactly. I mean, that's usually the efforts are end in December. So I wonder, because we're feeding past observation, I wonder if there was maybe not enough data for the model to accurately, pick up on those patterns. But that's just me speculating. I will not be able to answer your question.
What about the trend over time? It looked like, if you go back to your components of the GAM,
yes,
there was the trend over years, which is kind of interesting, and it at least looks like from that they're reaching maybe a plateau. I was curious what you make of that, the trend with respect to years.
The trend with years, yeah, I mean, so the mosquito is a invasive species to this area, and like you mentioned, reaching a plateau. So maybe that could be the mosquito finding equilibrium. so adjusting and adapting to this new climate. But I think there is still some work to do to to accurately say that yes, they're reaching equilibrium.
And then I see a question in the chat from Veronica Alba who's saying, being from San Bernardino County myself, I'm curious if there were any local forest fires or rain that might have added to these spikes along the trends.
On
So
the
that
trend.
affected the pattern in time, I guess.
It could be. I think we could also take into, we did not included fire. We did include precipitation, but like I mentioned, the water from the precipitation wasn't significant enough to, like we looked at the plot E right here, significant enough to observe, impactful effect on the abundance. But we didn't think of the impact of the fires. I think that will be interesting to take that into consideration for future work.
I know Dr. Attardo's group's thinking about biochar and its role in oviposition sites. I've wondered when we have a lot of smoke here in Davis sometimes, and I have wondered what mosquitoes do in those moments of high smoke coverage and
Yeah.
whether that also affects host-seeking behavior, for example.
Hmm. Yeah.
I don't know if anyone knows that. I'd be curious if anyone has comments on that or even anecdotal evidence. I haven't seen any particular studies that I can think of.
We haven't tried to study smoke because it's just so hard to kind of do experimentally, which is why we stuck with the biochar, but wouldn't surprise me if there's some kind of effect.
All right. Any other questions for Gaëlle? Okay, we'll keep it moving.
Thank you.
Thank you. Really nice talk, and look forward to seeing the end results of your modeling.
Thank you.
All right, so next up we have Dr. Geoffrey Attardo, who's known to many of you and is an associate professor in the Department of Entomology and Nematology here at UC Davis. He specializes in medical entomology, reproductive physiology, molecular biology, and genetics. His work centers on understanding the reproductive biology of insect vectors of human disease. The goal of the work is to develop a detailed understanding of the molecular biology and physiology of these insects and to exploit this information to control the insects and the diseases they transmit. The primary subjects of his work are mosquitoes and tsetse flies. He has studied how female mosquitoes regulate their reproductive cycle and gene expression in response to blood feeding, and demonstrated that mosquitoes require nutritional cues to begin developing eggs. And today we have the pleasure of hearing about Dr. Attardo's growing collaborations with some of our local Native American communities on wetlands. So over to you, Dr. Attardo.
Okay, great. Thanks everybody for coming and listening. This topic is a little out of my traditional wheelhouse, but I've been finding it to be very satisfying and kind of rewarding. So hopefully you'll find it interesting. So yeah, so I'm going to talk about today is some of the work we've been doing in wetlands restoration with indigenous partners in the Davis kind of woodland area. And I figured I would start with a land acknowledgement, as this land was occupied by Patwin tribes before Spanish settlement and Western expansion and the gold rush. So we acknowledge the land on which we're gathered. For thousands of years, this land has been the home of Patwin people. Today, there are 3 federally recognized Patwin tribes: the Kashil-Dehi Band of Winton Indians of the Calusa Indian Community, the Clutzel-Dehi Winton Nation, and the Yocha-Dehi Winton Nation. The Patwin people have remained committed to the stewardship of this land over many centuries. It's been cherished and protected as elders have instructed the young through generations. We're honored and grateful to be here today on their traditional lands. So one of the big aspects of this project is maintaining the transmission of that cultural knowledge, because much of that knowledge of the land and the ecology was lost during the time around the gold rush, when there were active attempts to eliminate these populations of native peoples. So one of the big priorities that we're working with is to try to provide opportunities for native youth to have exposure to traditional cultural practices around wetland maintenance, learning the language around these activities, and then learning the biology and the benefits of healthy wetlands. So my interest in wetlands comes from the fact that they've traditionally been associated with vector-borne diseases and other kind of just kind of negative impressions. And I was curious to understand that better. So wetlands have traditionally, in terms of Western society, been considered wastelands or dumping grounds, basically unusable land that was the only good use for it was to put things that we didn't really want to deal with. Overall, that it was unproductive and wasted space that needed to be reclaimed. It was typically associated with disease. So miasmas, going back to ancient Greece, danger, depression. In Christian traditions, it typically symbolizes stagnation and sin. We have a lot of it embedded in our language. So I'm swamped with work, I'm bogged down, I have to drain the swamp. There's a lot of popular culture that doesn't really help with our impressions of wetlands, including equating it with our sort of our government. So I was interested in learning more about this because the Patwin tribes have a very different relationship with wetlands than these kind of things would indicate. They were really dependent on wetlands for their livelihood, and they actively worked to maintain these spaces to ensure that they were productive and could help maintain their lifestyle. And really, what a lot of people are familiar with are degraded wetlands. So these are wetlands that are in a state of ecological imbalance or dysbiosis. And this is often a result of pollution, either in the form of industrial waste or agricultural waste or runoff. And oftentimes these waste products drive the ecosystem into disequilibrium. So you have overgrowth of bacteria and algae. This results in low dissolved oxygen levels, which results in the collapse of the food web. And really what happens is you end up with an overabundance of pathogenic microorganisms and an overabundance of species which are capable of living in these conditions, which many mosquitoes are able to live in these because they're not dependent on the oxygen in the water itself as they're breathing through a siphon. However, if you look at a healthy, well-balanced wetland, it really does the opposite, in that it acts as a source of clean water and a hub of biodiversity. So these are all animals that you can find up at Cache Creek wetland where we've been working. So we have active beavers, we have otters, lizards, a large array of amphibians and fish and insects. So it's a very healthy environment, which at one time had been an aggregate mining pit, which was not able to be used for agriculture, so it was turned over to the Cache Creek Conservancy for restoration. But biodiversity is a key property of healthy wetlands, and so they provide a diverse array of microbial, plant, and animal species. This results in competition for habitat with vector species. It also creates a dilution effect where you have alternative vertebrate hosts, which provide alternative blood sources, sometimes which are preferred over people, which results in reduced contact. And this was recently talked about in a 2021 paper called Anthropogenic Changes and Associated Impacts on Vector-Borne Diseases and Trends in Parasitology. But it really just talks about how the biodiversity, host diversity, and environmental health really helps sort of naturally control these things through competition, predation, and dilution. But as you sort of reduce the complexity of these habitats, you lose a lot of these control mechanisms, and you start to see sort of a refinement and selection for typically pest species like Aedes aegypti, which does particularly well in urban habitats. This is likely because there's not a lot of predators, primarily artificial habitats. Humans are the primary hosts, although Aegypti is really likes people. But you can see the pattern here. There was also a recent paper linking wetland ecosystem services with reduced vector-borne diseases in Puerto Rico. So they modeled neighborhoods and they in San Juan, and they found that neighborhoods that had were in proximity with greater coastal wetland area had significantly lower occurrences of dengue. This was after factoring in population density, income, age structure, and other environmental covariates. So protecting and restoring coastal and inland wetlands really offers nature-based multi-benefit strategies that are very low tech. So they function to mitigate climate change and acting as cooling urban heat islands. They're great buffers for floods and sea level rise. They enhance local biodiversity, and their sort of inherent ecological services also seem to reduce vector-borne disease risk. But this is also dependent on them being a healthy ecosystem and not degraded. And this is just kind of an example of that. So we were out in Med Lab last fall sampling for mosquitoes at the Yola bypass. So this is the bypass. This is the road here that leads up to it. And we parked here and we were sampling in the wetland here. And this is a video of where we were. So you can see there's lots of tule and cattail plants growing in here. And we had our students and people from SAC Yola Vector Control sampling, and I don't think we came up with a single mosquito larvae from the wetland area here. There's a predaceous diving beetle in there and some amphipods, which are detritivores. But literally across the street, we found these tire tracks that had accumulated water. And when you dip in those, you really see a different story. So you'll see in a second, it's just teeming with Culex tarsalis larvae and pupae. So I think it really highlights the importance of the ecological context in which mosquitoes are growing in as to their relative success. So I've been trying to understand, some of the mechanics of this and just get a basic hold on this. But there's a number of papers in the literature which are highlighting this, that the collapse of biodiversity, land use change, and the loss of the use of indigenous knowledge in helping to maintain these environments has really resulted in us living in our current ecological context isn't really what it was before colonization, and that we may be operating in a suboptimal state, which is due to a lot of the anthropogenic changes. So the wetland we're working in, so this used to be an aggregate mining pit. It's a palestrine wetland, which means it's freshwater. It's got a lot of reeds, sedges, cattails. It's non-tidal. It's a critical habitat for a lot of different animals, and it performs a number of essential ecological functions. And wetlands have been associated with groundwater recharge, erosion control, temperature regulation, water purification, carbon sequestration, biodiversity and animal habitats, nutrient cycling. They're great locations for doing biological education, recreation, and also there's evidence in the literature that spending time in these spaces actually improves mental health and other issues such as attention deficit disorder. In terms of carbon storage, they are the most successful ecosystem in terms of their ability to store carbon. They store 643 tons of carbon per hectare at a ground depth of 1 meter. And so you can see that soil, in terms of the amount of carbon stored, way outweighs all the other kind of sources. So having healthy soil and having wetland environments are really key to getting carbon dioxide back into the ground and out of the air. And unfortunately, California currently only has 5% of the wetland it did before the Spanish arrived here. So we're really operating at a severe deficit, these ecosystems. This is a groundwater table that looked at trends, and you can see that all throughout the Central Valley, groundwater levels are decreasing due to pumping for agriculture. Parts of the Central Valley have dropped literally by a foot due to the collapse of aquifers. So we really need to be working on groundwater restoration, and wetlands accomplish this by slowing the water down as it passes through the elaborate root systems of all these plants. And those root systems also provide channels that allow the water to permeate down through the sediment, through the ground and into the aquifers. And it's cleaned as it passes through these sort of filters on the way. So proper water management through healthy maintenance of wetlands is really important, as it reduces breeding sites. It helps recharge groundwater. It reduces water usage. The use of like native flora and plants in yards and landscaping reduces water use and also eliminates potential breeding sites generated due to requirements for watering. They improve biodiversity, which helps improve water quality, which I'll get into, and also generates a lot of predators which deal with pest species. And it also can reduce insecticide resistance. So the lower exposure of insects to insecticides and other aquatic contaminants like agricultural runoff or industrial runoff, suburban and urban environmental runoff, really lowers the kind of environmental pressures pushing on these xenobiotic detoxification mechanisms. So, well, how do these systems do this? So it's really about the plants, which function as the base of the food web, which are incorporating sunlight and carbon dioxide and converting them into cellulose and other nutrients. And these plants have these very elaborate root systems under the water and down in the sediment. And these roots, basically the plants have a symbiosis with the bacteria in the water, and they attract the correct bacterial species by secreting specific nutrients, which are called root exudates. Those root exudates attract the bacteria, which form biofilms on these roots. So what you have is a massive surface area throughout the entire wetland coated in this sticky biofilm that's covered in bacteria that have a tremendous abundance and diversity of biochemical enzymes which can degrade almost many of the compounds that are coming in as runoff. So these bacteria and the plants really form the foundation of the food web in the ecosystem. But if you don't have anything to keep the bacteria in check, they will overgrow, they'll use up all the oxygen, they'll pollute the water. And that's really where the other organisms in the wetland come from. So one of my students did a practicum project. We took water samples from the agricultural slough that feeds into the wetland. This is the wetland itself. So we collected from here, and then we collected from the outflow that the water leaves the wetland and goes into Cache Creek. And what we found was that in the slough, so these are just 6 samples from the slough, we see a greater diversity of bacteria, including these big orange bars are Pseudomonas bacteria, which tend to thrive in very nutrient-rich environments. We also see high levels of blue-green algae, which can be toxic if you've heard about toxic algal blooms. In the wetland, you start to see a decrease in these species as they either sediment out into the sediment, or they are eaten by the invertebrates living in the water. And then at the outflow, you can see you really have a refined microbiome where a lot of the stuff that came in has been sort of eliminated and incorporated into the biology of the wetland. In particular, Microcystis aeruginosa is what's been causing a lot of dog deaths and making people sick in association with algal blooms. And you can see here, specifically, the orange bars are Microcystis aeruginosa, and you can see in the slough, it's pretty high. It decreases by about halfway through the wetland, and by the time you get to the outflow, it's gone. So the wetland is really cleaning up the microbiome of the water and kind of refining it. So we've been, part of our workshops have been to use microscopes and water samples to examine what organisms are living in the water and to introduce people to a whole area of life that they just had no idea was actually there. So this is a microscopy image of some of the bacteria that we get out of the wetland. So all of these organisms you're going to see in these videos came straight out of the Cache Creek wetland. So these bacteria are in the water, and to maintain their population, you have a number of single-celled ciliate microorganisms, which are sort of the next level up. So these are Vorticella, and they are attached to a bacterial biofilm that looks, it's this sort of gelatinous stuff, and you can see all the bacteria squirming in it. So these guys attach themselves, and they basically filter the water around the biofilm and are ingesting any stray bacteria that come loose. So they're helping to regulate the population numbers of the bacteria. We also have large numbers of paramecium, which are also great filter feeders. They're coated in cilia, and they basically, you can see this one, all the particles that are flying by. It's basically putting them into its food. It's basically like a mouth where it fills up these food vacuoles, which are filled with digestive enzymes and break down bacteria and detritus that are in the water. So you can see these are much larger numbers of paramecium, as well as these purple trumpet-looking things are called stentors, which are the largest single-celled organisms found. They also have a trumpet-like opening, which is surrounded by cilia, which they also generate a vortex which pulls water in, as well as any bacteria or detritus. So all of these little basically invisible microorganisms are working 24/7 to clean up any stray nutrients that are floating around in the water. This is a picture of a bloom of paramecium, which are responding to, so there was an addition of nutrients into this tank that I was keeping. The bacteria started to bloom, and then the paramecium bloom in response. And you can see them following chemical gradients to high concentrations of bacteria. They almost look like schooling fish. So all these microorganisms form the base, and then on top of that, you have more complex organisms. So you have a lot of different worms, crustaceans like these zooplankton, Daphnia, amphipods, which are relatives to shrimp. This is a filamentous fungus. So you can see the kind of bizarre and cool organisms that are occupying this space. So there are also a lot of snails, which are big algae eaters. This is called a bladder snail. They're able to regulate their position in the water column using air. This worm we haven't identified yet. I've sent it to some nematologists and they haven't been able to figure out, but it's pretty dynamic. These are tubifex worms. They're called sludge worms. They're very common, but they help turn over the sediment and keep the nutrients that are in the anaerobic area up above, allowing the other organisms to feed on them. And we think that this worm is doing something similar, but it's pretty unusual. This is a piece of biochar I've put in the water, and you can see that all these different organisms really like hanging out on the biochar. And there's a lot of evidence to suggest that biochar does good things for soil and good things for water quality, and these animals seem to kind of reflect that. So we've got copepods, we have seed shrimp, we've got amphipods, we've got multiple types of oligochaete worms, snails. So these are all bacteriivores and detritivores that spend their time 24/7 just cleaning the water. So then you do have mosquitoes in these spaces. So our kind of native species like Tarsalis typically breed in stagnant kind of standing water. They prefer to feed on birds and occasionally on mammals, but there are primary West Nile vectors around here. So they're moving the viruses between the birds and humans and animals. And one of the other things about wetlands is they are a big source of bird population. So there is opportunities for acquisition of viruses from those sites, but there's also more birds there, giving the mosquitoes their preferred host. So I don't entirely understand the dynamics of that, but I think the more wetland area you have, the more potential possibilities for dilution occur, rather than concentrating it all around individual water sources. So these guys work well on the stagnant water because they've got these long siphons that allow them to breathe atmospheric air, which a lot of the other organisms in the food web are dependent on dissolved oxygen. So if the water quality drops, a lot of those other organisms die and you lose that competition. But the mosquitoes are also acting as filter feeders, similar to these other organisms, in that they're creating these currents. They're pulling in bacteria and detritus and incorporating it as nutrients. But that also makes these mosquitoes a popular source of nutrition for other organisms. So pretty much in every sample I take, we find these copepods, which are these little crustaceans, and they're small, but they're pretty fierce. And there's been a significant amount of research showing that they're actually pretty effective at controlling Aedes larvae. Typically first or second instar larvae, because these copepods are small, but they're very abundant and they reproduce quickly and they're relatively easy to rear. So they can be, just added to potential breeding sites as a competitor in urban settings. Yeah, there was a talk at MVCAC. The keynote speaker, I think, was working on this, Neil Lobos. We also have a variety of other predators. This is called a back swimmer. It's currently double fisting. It's got a copepod in one hand and a Daphnia in the other. But these guys are very aggressive predators and will go after mosquito larvae just as well. These are bugs, so they use their beak. You can see it's inserting its beak into the Daphnia there, and they inject them with digestive juices and then kind of suck out the insides like a shake. This is a diticid beetle larvae. These are also very fierce predators. If you watch this Daphnia right here, you'll see that it's about to have a bad day. But these guys are also voracious eaters. And they're really designed to be sort of exclusively predators. You can see it's chewing up that Daphnia pretty good, but they also, both the larvae and the adults of these are very successful predators. We also have large numbers of dragonfly larvae, which are dragonflies are predators both in their larval forms and in their adult forms out of the water, and they're pretty successful in both situations. So they have a lower lip that extends out, and they're able to catch their prey either by chasing it down or by waiting, disguising themselves in the sediment and waiting for prey to come by. This is a larva that I brought into the lab, and I added some Aedes aegypti larvae to the tank, and you'll see that it just gobbles them down. It's so fast, it's really hard to get on camera, but you can see it went through about 40 larvae in about 10 minutes. So they're very voracious and aggressive. And then adult dragonflies are extremely good flyers, and they have about a 95% success rate when they go after prey, which is better than any other predator, and that's known in the world. There's also lots of damselflies, which are relatives to these guys and also predators. We have western fence lizards, which are insectivores and also have the beryllicidal factor in their blood, which helps clear ticks. We also have large numbers of cliff swallows at Cache Creek. So these are all birds that make their nests here, and they spend all day just collecting insects for to feed their offspring. So this is a very big colony, which suggests that they're doing very well on the nutrients that are in the form of insects that are coming out of the water. So I've been pairing up with my partner here, Diana Almendarez. She's a Winton Patwin cultural practitioner and ethnobotanist. So she has a deep knowledge of all the plants and animals around the wetland and how they were used, as well as she's a basket weaver and a dancer and knows all of that. All this information that she learned from her grandmother, who lived to 103 and lived from, I think, 1898 to just past the year 2000. So she saw a tremendous amount of changes. But so in our workshops, she's been teaching cultural burning. So that helps with carbon sequestration. It converts plant matter into biochar and removes it from the fuel cycle. That helps enrich the soil and also promotes ecological restoration by killing off non-fire tolerant invasive flora and promoting fire dependent or tolerant flora. So this is a burn we did on Cache Creek on May 14th. And then so you can see the kids here. These kids are all from of native descent, from local tribal groups, and they're learning how to safely do burns. And CAL FIRE is integrated into these activities, so they're always there and watching and learning. But this is the same location 5 weeks later, and you can see these are tule plants. So it really actually benefits from the burn, and it comes back very quickly. And tule is really a staple that was used to make clothing and shelter and houses by the local natives. They were really dependent on this as a resource. It's also edible. You can make a boat from it. It's extremely buoyant. So it's a really versatile and great sort of working tool. So our workshops we did last summer, we were collecting cattail and tule from wetlands. We then took all of that material we collected and dried it and prepared it for weaving. And then in the workshops, we wove with the tule into mats, basically. And the purpose of the mats is to seed new cattail into parts of the wetland that need additional plants. So we've made these mats. You can weave the cattail roots in, and then they act as a platform to establish new plant life. And then we attach them to a cordage or rope that we made out of cattail root or cattail leaves, which is extremely strong. It's really impressive. So this is sort of our final product from the workshop, and then we actually put it out into the water and just observed it gets rapidly colonized by invertebrates and other organisms. And it also helps break down algae because you help boost the invertebrate populations with this food. So if you're interested in reading more detail, we had an article recently published by Bay Nature that goes into a little more detail on the project. But yeah, so that's that for my talk. I've got a lot of other stuff associated with this, but I think it gives you a pretty good overview. So I'd really like to thank Diana, without whom I would never be able to do any of this. And the members of YOLO Sol, Juliet Beck and Anuj Vaida, who have been helping support Diana and these initiatives, as well as Lynn Harrelson and Jolene Gindrich at Cache Creek Conservancy, who help us out, and my grad students, Nicole and Cece, who participate in some of these activities, and especially for the CDC and PacVec for being willing to support kind of an out of the box type initiative like this. So thanks.
All right, thank you, Dr. Attardo. That was great, and every time I see you present anything related to this, it grows, and it's clear you might have a second career as a National Geographic videographer. Pretty amazing stuff. All right, with that, I know we're past the top of the hour, but I want to open up any opportunity. You can either unmute yourself or write in the chat if you have any questions. All right, I'm going to ask one really quickly, and I was just curious about the permanence of water because I just thinking about mosquitoes specifically, predators and other aquatic insects take over water pretty quickly when it persists, and mosquitoes have long made their living by taking advantage of intermittent water sources that don't let the predators catch up. And I'm wondering purely from a water quality standpoint, if you take a really nice wetland versus another persistent water body, what are the differences in terms of mosquitoes?
So like the other persistent water body would be like I don't know, like just a unmanaged pond or something like that.
Yeah, or a ditch, or just anything that holds water for a very long time and allows predator populations to build.
Yeah, I mean it depends on the dissolved oxygen levels in the water. So a lot of those predators don't do well in poor water quality. So like I've had a couple samples where I was experimenting by adding nutrients in, and when the bacteria grow too quickly, they overwhelm the capacity of the other aspects of the food chain to compensate, and then the oxygen level basically tanks, and then like the dragonfly larvae all died. Um, the the diving beetle that I had died. So it seems like there's the water quality in a wetland that's sort of like where there's a balance is much better. The water's actually there's pretty much any nutrients that come in are utilized very quickly. But if you don't have that sort of structure in there, a lot of those nutrients just are floating around or either producing more bacterial um, uh, numbers, or mosquitoes can really take advantage of that as well. So, but oftentimes the difference you'll see is healthy plant life around. The plants are really what are sort of driving the structure with the root exudates and all of that.
A really great view of the complexity involved. Really appreciate the talk.
Yeah, thanks.
All right, so I know we're a little past time. I just want to especially thank Jenny Nguyen, who's I think still on here somewhere. But Jenny has really done a great job of getting these seminars organized this year and also communicating with all of us to keep us all aware of when they're happening. This is our final seminar of the year, so we will be having the seminar series again starting in fall. So if you have ideas for future seminars, please feel free to reach out to us. You can send a note to questions@pacvec.us, or you can send a note to Jenny or me, and we'll try to accommodate those however we can in the fall series. And with that, I just want to thank Dr. Attardo and thank Gaëlle for your talks today and for all of you for attending. We'll look forward to seeing you again in the fall, if not before.
Yeah, thank you, Chris.
Thank you, Chris.
All right. Thank you all. Have a good afternoon.
Bye, everybody.
Have a great afternoon, everyone. Bye.